In order to understand the mechanisms involved in the transport of substances in plants, we must first look at the structure of the root and the stem and relate this to the function of transport. We start by looking at the external features of the root. Now, we start by looking at the root part of the plant, and the first part of the root that you're going to look at are the external features. The external features of the plant. Now, the roots are structured, particularly structured to perform their primary functions. Now, there are two types of root system depending on whether the plant is dicotyledonous or monocotyledonous plant. In these diagrams here, you have the taproot system, mainly found among the dicotyledonous plant. And in this type of root system, the primary root or the radicle remains dominant. This is the primary root and grows straight downward. This is the taproot, and branching from it are smaller lateral roots. So this is a taproot system consisting of a dominant taproot that grows downward, and then lateral roots, many lateral roots that branch from it, and even smaller roots branching from the lateral roots to form this taproot system. The second one is the taproot system found among the monocotyledonous plants. In this system, in addition to the primary root, that is the radicle, several secondary roots, all of which are about the same size as the primary, also arise. So that we have a fibrous root system consisting of many roots of about the same size spreading from the base of the plant to the surroundings. So whether it is the taproot system or the fibrous root system, the root of a plant is adapted to perform the following primary function. First is anchorage. The roots, because of their extensive branching, that provide support to the plant by holding it firmly in the soil. This branching out increases the base size, and in that way the plant can stand firmly in the soil. Secondly, the roots are the site through which water and mineral salts are absorbed into the plant. So this great branching also helps to increase the surface area. The branching increases the surface area of which water and mineral salts can be absorbed from the soil or any other surrounding medium into the plant. Now, the other functions performed by certain specialized roots in certain plants, and these include storage. For example, in plants like cassava, the roots become swollen with food reserves. So that these roots not only play the primary roles of anchorage, absorption, but also of storage of food substances. Then there is also, in other plants, roots playing the function of gases exchange site. In plants that grow in swampy areas where the soils are waterlogged, there are those specialized roots that grow out of the soil and they are involved in gaseous exchange, and for this reason these specialized roots are also known as the breathing roots. So they grow out of the swampy, waterlogged soil and into the atmosphere, where they enable the plant to exchange gases with the surrounding, and that is mainly taking oxygen and release carbon dioxide to the surrounding. Now, looking at the internal structure of the root, first you look at the longitudinal section. A longitudinal section is made by cutting along the length of the root, and when that is done, the following features are observable. Projecting from the epidermis are extensions known as root hairs. Then you have the epidermis, that is the outermost layer of cells. Then immediately below the epidermis you have the phloem, and then the xylem at the center. Towards the tip of the root, there's a specialized region known as the apical meristem. This apical meristem consists of undifferentiated cells which actively divide to give rise to new cells that become tissues and therefore contribute to the increase in length of the plant. At the terminal end of the root, there is a root cap. The root cap consists of simple parenchyma cells that protect the apical meristem as the root tip pushes past the soil particles. So the root cap is a protective structure that is relatively impermeable to water and solids. So as the outer cells wear out and die out, new cells from the apical meristems are produced to replace those that are worn out and fall off. Now, looking at the transverse section. Now, a transverse section is made by, say, cutting across the length of the plant root, and when that is done, the following tissues are observable. Now, the tissues that are present in both the monocotyledonous plant and dicotyledonous plants, the epidermis, there is a cortex, phloem and xylem, and endodermis. But the arrangement and the pattern of distribution varies depending on whether it is a dicotyledonous root or monocotyledonous root. As this is shown clearly in these two diagrams here, you see in the dicotyledonous root, the xylem occupies the central position, forming a star-shaped structure. Then in between the arms of the star is a phloem tissue. Surrounding the xylem and the phloem are two rings. The innermost ring is the pericycle. Then to the outside of the pericycle, the endodermis. Then after the endodermis, there is a cortex made up of parenchyma cells. The cortex extends outward and forms or joins the epidermis. So after the cortex, there is the epidermis that forms the outermost layer. In the monocotyledonous root, the epidermis is the outermost layer. Then beneath the epidermis, there is a cortex made up of parenchyma cells. Then there is a ring of specialized parenchyma cells making up the endodermis. Then inside the ring, there is phloem alternating with the xylem, arranged in a circular pattern. Then at the center of the vascular bundle, there is a pith made up of parenchyma cells. So, similar tissues are present in both the dicotyledonous and monocotyledonous root, but the pattern of arrangement differs, and on that basis, you can differentiate between the two types of plants. Now, let's talk a little bit more about the type of tissues that are present. The type of tissues that are present in the roots of plants. Starting with the epidermis. Now, as mentioned earlier, the epidermis is the outermost layer of cells, the outermost tissue that serves the function of protection. It protects the underlying tissues. In certain regions of the epidermis, especially just behind the tip, there are specialized epidermal cells known as the root hair cells. Root hair cells have an extension known as the root hair. Now, the root hair greatly increases the surface area. So that is the root hair of the root hair cell. So this greatly increases the surface area for absorption of water and mineral salts into the plant. So through the extended root hair, water and mineral get absorbed into the roots. Now, this region of the epidermis where the root hair cells are present is known as the piliferous layer. It's known as the piliferous layer. And the cells, that is the root hair cells, are thin-walled to allow easy passage of water and mineral salts. Now, as the root tissues mature, these root hair cells lose their root hair, they become less permeable, and they assume their protective function. So they become rather protective instead of absorptive. So the epidermal cells now become less permeable and they become protective. So for most part of this root, the epidermis consists of protective epidermal cells. But in regions just behind the tip, there is a piliferous layer consisting of root hair cells with root hair. The root hairs provide a large surface area for the absorption of water and mineral salts. Then in the root, you also have parenchyma cells. Now, parenchyma cells consist of thin-walled cells. Thin-walled cells that also have relatively large subvacuole, and these thin-walled cells or parenchyma cells, they play the role of packing and filling spaces between other tissues within the root. The parenchyma cells that are found just beneath the epidermis, that is between the epidermis and the vascular tissues, make up the cortex. while those that are found within or enclosed by the vascular bundle ring make up the pith. So cortex and pith consist of parenchyma cells. The name depends on the location. If they're found at the center, then they make up the pith, but towards the epidermis, then they make up the cortex. Now, apart from filling up the spaces between the other tissues, the cortex and the pith also act as storage. Within these cells, they can store a lot of water and food substances. Then the endodermis. The endodermis consists of a single layer of cells that surrounds the vascular bundles. The endodermal cells are characterized by the following features. 1. They have starch grains. There are numerous starch grains. And then secondly, there is a woody band of material on the outer surface. A woody band of material on the outer surface known as the Casparian strip. The Casparian strip. This Casparian strip is impervious to water, and it enables the endodermis to control the amount of water and mineral salts that enters into the vascular tissue, and it also helps, or it ensures that the water coming into the vascular tissue passes through the cytoplasm, because it being impervious to water, it ensures that the water is deflected into the cytoplasm, all the water that goes towards the epidermis, and within the cytoplasm, any harmful materials like pathogens or excess minerals can be screened out. So the endodermis play that dual role of 1, controlling the amount of water and mineral salts entering into the vascular bundles, and secondly, it also screens out unwanted materials that may be present in the absorbed water, such as the pathogens. Then the pericycle. The pericycle is a single layer of cells that located immediately below the endodermis. It's responsible for giving rise to lateral roots, the branches of roots that come off the taproot. So pericycle is present in the dicotyledonous root, but it is absent in the monocotyledonous roots. Then the xylem and phloem making up the vascular tissue. And starting with the xylem. The xylem consists of 2 different types of dead cells. There is the vessel elements and the trachea elements. Both vessels and trachea elements have thickened lignified walls that enable them to withstand high pressure without collapsing, and also enable them to serve as supportive structures to the plant. So both of them have thickened lignified walls for strength and also to prevent them from collapsing, because they both have hollow lumen. Both of them have hollow lumen for free passage of water and mineral salts. Now, in both the vessel and the elements, the end wall, because the xylem element consists of elongated cells. Now, these elongated cells consist of side wall and the end wall. Now, in both vessel and trachea elements, the end walls are either partially or completely broken down to allow for free passage of water and mineral salts from one element to the other. So these end walls will either be completely broken down or will be partially broken down to allow for the free movement of water and mineral salts from one element to the next. The elements are made up of dead cells, and thus they cannot utilize the water and mineral salts that they conduct. And also, they all have bordered pits. Bordered pits are pores, tiny pores on the side walls, perforations on the side walls that permit lateral movement of water to the cells surrounding the elements. So those are the features that are common to both the vessels and the trachea elements. However, there are some important structural differences between the two. The first of them is that whereas the vessel is circular in cross-section, the tracheids would usually be 5 or 6-sided in cross-section to have an angular cross-section. Secondly, in the vessel, the end walls are completely broken down so that there is a free flow of water and mineral salts from one element to the other. But in the tracheids, the end walls are present and perforated, so water and mineral salts must pass through the pores. So this considerably slows down the rate of passage. So in relative terms, the efficiency of the movement of water in the vessels is much higher compared to that of the tracheids, because in the tracheids there are end walls which are perforated, but in the vessels the end walls are completely broken down to allow for the free passage of water and mineral salts. Now, the vessels also are cylindrical. Vessels are cylindrical in shape, while the trachea elements are tapering. They're tapering towards the end. So those are the very distinct structural differences between the tracheids and the vessels. However, they both occur. They both make up the xylem element, and they're found in flowering plants. That is, both the vessel element and the trachea elements are present in flowering plants, making up the xylem tissue. Now, the phloem. The phloem is part of the vascular bundle. It is made up of two types of cells: the sieve element and the companion cell. The sieve element contains numerous cytoplasmic strands that run through it and extend from one element to the next. It is these cytoplasmic strands that play a role in translocation of substances from one element to the other. So the cytoplasmic strands are continuous through the sieve pores. This is the end walls that separate one element from the next. So you have the sieve element. At the end, there is a sieve plate. Each sieve plate has perforations known as the sieve pores. So the cytoplasmic strands pass through the sieve pores from one element to the next. So this facilitates the movement of substances from one element to the other along the cytoplasmic strands that pass through the sieve pores. The sieve elements have no nucleus and they have very few organelles because of their thin cytoplasm. On the other hand, the companion cell has a prominent nucleus and many organelles. For example, it has numerous mitochondria. The function of the companion cell is to control the activities in the sieve element. So basically, companion cell controls the translocation function of the sieve element, and it also provides energy that is used in translocation, because translocation is an energy consuming process. The sieve element does not have enough mitochondria on its own, since it is mainly filled with cytoplasmic strands. So it relies on the companion cell to provide it with energy for the process of